Purines have two nitrogen bases, such as adenine and guanine. Pyrimidines have three nitrogen bases, such as cytosine, uracil, and thymine. DNA nucleotides consist of one of four bases: guanine, adenine, thymine, and cytosine.
Nitrogenous bases. The bases in DNA are adenine, guanine, thymine, and cytosine. In RNA, the nitrogenous bases are uracil, guanine, cytosine, and adenine. DNA polymerase I. This moves RNA primers and replaces them with DNA nucleotides. DNA polymerase III. This adds DNA nucleotides that continues on the leading strand. Double helix is a long and linear molecule made up of smaller units called nucleotides that form a chain. The DNA backbone is made by altering phosphate and sugar residues held together by phosphate groups. Phosphodiester bonds, covalent bonds. One of these comes in ribose found in RNA. It is a phosphate connected to ribose sugar, connected to a nitrogenous base. In this case, is connected to UCL. Hydrogen bond. Hydrogen bonds are weak chemical bonds between an electronegative atom, such as oxygen and hydrogen. They are responsible for the properties of H2O.
Okay, one of these is called the leading strand, and the other one is called the lagging strand. The leading strand is replicated continuously in the 3 to 5 direction, and the lagging strand is replicated discontinuously in short sections. These sections are called Okazaki fragments, and they are short lengths of DNA.
When the DNA polymerase complex is making DNA, it moves alongside the template strand in the 3 to 5 direction. Since DNA is antiparallel, the new strand is synthesized to the 5 to 3 direction.
Topoisomerases are enzymes that participate in the overwinding or underwinding of DNA. During DNA replication and transcription, DNA becomes overwound ahead of a replication fork.
Single-strand binding proteins bind near the replication fork and holds DNA apart so hydrogen bonds don't reform. DNA helicase is an enzyme that unzips DNA and breaks up the hydrogen bonds between nitrogenous.
DNA copies itself. The two strands of DNA molecule have complementary base pairs. The nucleotide sequence of each strand automatically supplies the information needed to produce its partner.
DNA helicase is an enzyme that unzips DNA and breaks up the hydrogen bonds between nitrogenous.
A primer is a short section of RNA.
DNA ligase, or the glue, joins the DNA or the Okazaki fragments.